So we just presented a preclinical data showing the efficacy of two antibodies, anti-PD-1 and elotuzumab, in mouse tumor models. And the rationale for the study is that elotuzumab is an approved agent for treatment of multiple myeloma, and it is a tumor-targeting antibody, and it works by using cells of the innate immune system, and it kills myeloma cells via a mechanism called ADCC, which stands for antibody-dependent cellular cytotoxicity. And the second agent that we were studying was anti-PD-1. And PD-1 is a checkpoint receptor. It's an inhibitory receptor that's been characterized to regulate adaptive immune cell response to the tumor. And a lot of data, recent data from both preclinical studies as well as more recently clinical trials, have showed the efficacy of anti-PD-1 blocking antibody in multiple solid malignancies, and more recently in hematologic malignancy in Hodgkin lymphoma. And so the rationale was that if we combine two agents that work on two different arms of the immune system, the innate immune arm and an adaptive immune arm will be able to elicit a better anti-tumor response. And this is what we observed in our mouse models. When we combined elotuzumab with anti-PD-1 blocking antibody, we saw an enhanced anti-tumor response. Tumors regressed, was increased mouse survival, and the more mechanistic studies have shown that this is due to infiltration and increased activation of both cells of the innate and adaptive immune system. So we're sort of taking two arms and then increase them simultaneously. And so this preclinical data from mouse studies really provided us with the rationale to continue and test this combination of elotuzumab and anti-PD-1 in patients with multiple myeloma. And that's indeed the trial that's just been initiated.
Okay. Just speaking about some of the mechanisms there, could you talk more about the elotuzumab binding pathways?
Yes, so it binds to a receptor that's overexpressed on myeloma cells, and that's one of the mechanisms by which it kills myeloma cells. And it also, SLAMF7, which is its target, is also expressed on NK cells themselves, so it activates these cells simultaneously.
And you've addressed the outcomes and the ongoing future trials. Is that still recruiting for patient cohort?
Yes, so it's just begun, I believe, in March, recruitment. So it's just beginning. Yes, so it's an incredibly exciting time, and there is definitely preclinical data suggesting for the role of PD-1 pathway in multiple myeloma. So I guess time will tell how it works in patients, but data from solid tumors particularly and Hodgkin lymphoma recently have definitely shown that it is capable of improving an immune response to tumors. So hopefully we'll do the same in the context of multiple myeloma.